like many of the other speakers, I'm also talking about something a little bit different today. So I'm going to talk about a sensor platform that was developed in my lab called corona phase molecular recognition in order to identify and sense arbitrary small molecules. So I apologize a little bit to anyone who's doing chemical engineering or material science. I'll try to go over a bit of a background on the sensors we're using. But first, I just wanted to make a note. So my situation is a little bit different from most of the CSGF fellows in that I primarily work in a large experimental research group. So you can see most of the members of my lab, it's about 20 to 30 people. I'm the only one who does simulations all the time. There are a few people that I'm really hoping are going to do simulations in the future, but hasn't been determined yet. I'm pushing. And so I'm split between two groups. One is the Strano group, which does experimental devices on low-dimensional carbon materials, and I'll describe what I mean by that. And the Bratz group, which does systems engineering, controls, and applied math to help with the simulations. So what do I mean by low-dimensional carbon materials? These have really come up in the past 20 years, not because they weren't made in the past, but just because it's been very difficult to isolate and study them. So in the mid-1980s, it was C60, Buckminster fullerene, was discovered, and that led to the Nobel Prize in Chemistry for its synthesis in 1996. And then in 1991, single-walled carbon nanotubes were discovered. And then even more recently, graphite, which is composed of individual layers of graphene, they finally came up with an easy way to isolate single layers and study them. So this led to the Nobel Prize in Physics in 2010. So there's been a lot of excitement and a lot of interest in these materials, primarily because the confinement that you get when a single sheet of carbon is rolled up into a ball or a tube, or you look at a single layer, leads to very interesting electronic structures. And so it's different from if you just had a single sheet of graphene, or if you were just looking at the electronic properties of graphite. So we use carbon nanotubes in our lab primarily because they have a very strong fluorescence signal. So what's happening is when we excite with light, we excite electrons from the second valence band to the second conduction band. And then this electron hole pair relaxes to the first conduction band and the first valence band. And at that point, if the electron and hole recombine, then you can get emission with light coming out. Or if there are defects on the surface or other pathways, you can have recombination without emission. And so there's a very strong fluorescence signal. We can see individual molecules in a microscope. This lends to single molecule studies. And if you have defects on the surface, you can reduce this fluorescence intensity. And depending on the structure of the carbon nanotube, exactly how you roll up these graphene surfaces, you can adjust whether it's a metal or a semiconductor, and also the positions of these bands, the E22 and the E11. And so when people talk about, people say that they're using carbon nanotubes, it's very important to keep in mind that there's a wide range, and you can tune the electronic properties. So if we just look at the E22 and the E11 emission wavelengths and plot them for all the different types of species, we can see that each of these has a different pair. And so we can choose which species we want for a particular absorption window for whatever experiment we're trying to do. So we use these carbon nanotubes and the fluorescence signal to detect small molecules. And the way that we do that is we wrap the carbon nanotubes in a polymer or a wrapping of interest. As an example, I'll show DNA, AT15 repeating. And so the experimental setup we have is carbon nanotubes on a Petri dish in solution, and we excite with a 685 nanometer laser, and we look at the fluorescence emission out. And here's a little picture of what we can see in a microscope. We can see a little pixelated dot. That's a single nanotube fluorescing. And depending on the concentration of the small molecule that we use, in this case DNA AT15 recognizes nitric oxide, we can actually see these individual quenching events, which are individual nitric oxide molecules adsorbing to the surface of the nanotube and quenching the signal. So this is a single molecule study that we can see very easily in a microscope. And of course, depending on the concentration of nitric oxide, we can either get a very quick quenching or a very slow quenching. And so if we look at the rate at which these events happen, we can get an idea of what the nitric oxide concentration is local to the carbon nanotube. So this is an interesting problem because there are many important effects at this scale. So the chemistry of the carbon nanotube surface and the polymer is very important. The electronic structure is obviously what we're actually seeing, the fluorescence intensity. The local environment changes the electronic structure of the carbon nanotubes, so you can get solvatochromatic shifts. And if you strain the carbon nanotube, you can also change its electronic structure. So the geometry and the physical structure interplays with the electronic one. And at the same time, sort of the scales that we're interested in for electronics, the unit cell might be 200 to 500 atoms. We can do that pretty easily with tight binding density functional theory, sort of like on the order of a day with a modern desktop. And if we're looking at how, for example, one of these polymers will wrap a carbon nanotube, if we include like a small solution box, it's usually about 100,000 atoms for a single carbon nanotube unit cell. And so that's something that is relatively straightforward to do with molecular dynamics without having to go to the limits of what people are doing for other systems. So an interesting question is how exactly these wrappings lend themselves to analyte specificity. So in the past, there have been a large number of experiments done within my lab to show or to screen different wrappings like DNA or these other polymers that I'll talk about to see what they detect. And so we found experimentally that DNA recognizes nitric oxide preferentially. But we also found that these other polymers, rhodamine isothiocyanate connected with a polyethylene glycol chain, happens to recognize estradiol, and FMOC PPEG8, another arbitrary polymer that one of the graduate students picked, happens to recognize L-thyroxine. And so this was very interesting in that they were able to screen for all of these and try a lot of polymers and check against a lot of analytes, but there wasn't really an understanding of what was driving these interactions and what lent themselves to the specificity. So to do that, we wanted to model what was actually happening near the carbon nanotube surface. So for the case of Ritzy-PEG-Ritzy, there are these hydrophobic Ritzy molecules on the end connected with a long PEG backbone. And so if we step back a little bit, this sort of looks like two hydrophobic anchors connected with this PEG chain that will stick out in the solution. And this is a coarse-grained Martini simulation of the same system. And we can see in blue these Ritzy molecules sort of stacked up against the green carbon nanotube surface. One is extending out a little bit. And the hydrophilic PEG chains are extending out into solution. And so if what we're interested in is how small molecules can get into this area and interact with the carbon nanotube surface, then the most important aspect is how are these hydrophobic molecules stacking against the surface, and that's really all we care about. So the simplified picture that we want is what does this corona phase look like, the phase that's actually right next to the carbon nanotube surface. So this is something that we can model fairly easily if as a competitive adsorption process between the hydrophobic anchors and small molecules that are trying to get to the carbon nanotube surface. So if we have some idea of what the partition energy is between solution and the carbon nanotube surface for each of these species, and how they interact via van der Waals, the original mole fraction in solution, and the mole fraction, we can then calculate the mole fraction on the surface. And so we sort of have this hard sphere model where these two species are competing. So the difficult step is, of course, calculating things like adsorption energy for every one of these species. But thanks to other groups, specifically the the drug screening community, it's relatively straightforward to get at least very coarse force field parameters for arbitrary small molecules. And so we can do a simulation where we look at the adsorption of a small molecule to a graphene sheet. This is neglecting the curvature of the nanotube. Calculate the adsorption energy for different molecules, and then feed that back into the thermodynamic model that I showed on the previous page. So if we do that for all of the molecules, we see that we get what we define as agreement within 20%, which is about what we think the error bounds on our experiments are for about 83% of the experiments. And what we also found was that if we pre-screened with this model, we would be able to increase the success rate of these future experiments from about 21% to about 40%. So that would obviously save a lot of the poor graduate students' times in my lab, rather than just testing obvious combinations that are probably not going to work out. So the main idea is try to screen out all of these that aren't going to work very well. So there are many aspects to this problem. So the one I talked about was how do these wrappings lend themselves to specificity, but we also want to actually apply these to actual uses. So we've also taken these DNA wrappings and started putting them into cells to do in vivo sensing, and so I'll just talk a little bit about that. So what we found is that we can localize, when we introduce these DNA-wrapped carbon nanotubes to a single A375 melanoma cell, we can see that the carbon nanotubes are sort of localized around the lysosome, which is stained with lysotracker red. If we use a very similar setup where we have a cell and we're exciting and measuring the fluorescence signal, we can actually look at the nitric oxide concentration or the intensity of the carbon nanotube fluorescence over time and get spatiotemporal imaging within a single cell. So we're trying to extend this to the study of other molecules within cells, and then also other members of my lab are also doing animal studies at the moment to figure out how to apply those to rats and other small animals. And this is just an integrated intensity over time. So you can see over the course of about 6 minutes after we introduce a nitric oxide producing drug, about 10% of the nanotube signal is quenched. So that's something that we can see very easily experimentally. So with that, I would like to thank the CSGF Fellowship and NSF and Exceed for resources. It's really amazing how easy it is to get computational time. That's something I didn't really understand before I started this program. My group doesn't have any computational resources. We don't have any clusters. But writing my own grants for Exceed time, it was possible to do all the simulations for this work. And so it was nice being able to come in, see how to put the grants together, talk to people who said they'd done it before, and then just go ahead and get the time. And then if I was the one writing the grants for the time, I could just ask for whatever I wanted to make sure I had what I needed. And I also wanted to say that the CSGF program has really influenced my PhD in other ways as well. So one of my advisors is also the same advisor of Ashley Ford, so I knew her coming in, and he already knew what to expect from a CSGF fellow. And even one of my committee members, Martin Brizant, graduated from the CSGF program in 1996. So it's nice seeing so many people around and sort of feeling like there's a small community among the simulation and modeling people just from being in the fellowship. And of course, all of this work is is done with the help of many experimentalists. So there's a large subgroup of people working the nitric oxide and single molecule subgroups, and also the nanopore subgroup. So with that, I'd like to take any questions.